Good afternoon. Welcome, and delighted to see so many people come to hear our presentation about Pfizer. My name is Enoch Huang. I lead a team called Machine Learning and Computational Sciences within Pfizer R&D. I'm joined with 4 of my colleagues and teammates to tell you about some of the work, but I'm here to just introduce the department and to give you a high-level overview of who we are. So this department was actually started back in 2007. Like many pharmaceutical companies, we are organized by disease areas and function. For example, we have a group on oncology research and with people in medicinal chemistry who design and synthesize compounds. We are the single largest computational sciences group within Pfizer R&D, and we partner with support all of these embedded computational scientists, those that work on target selection for new projects for our portfolio, as well as people who design and optimize molecules, both large molecules and small molecules. And so our specific role is to develop computational tools and algorithms and methods for practitioners throughout the Pfizer R&D organization to use. It's a privilege to be specializing in method development because of the, as you know, the technology in the field changes so rapidly. And as you'll hear from some of the more recent project examples, the focus on machine learning research is a very strong at the moment with applications in these 4 areas that you see around target selection and indication prioritization, molecule design, understanding disease heterogeneity and pathophysiology and so forth. Here are some examples of work that we've historically done. I won't go through these examples in details because you'll hear examples from my colleagues in just a moment, just to give you the impression that we work both in the biology and the chemistry domains. We tend to specialize in high dimensional data, which you've heard many examples earlier today, NGS data, genetics data, mass spectrometry data, just to use some examples. We also have a strong technology focus on scientific computing, both HPC and cloud, that supports everything we do in our group. One question I get a lot is where are we based? And so one reason why we're excited to be here in Lyon is because we have a growing European footprint. MLCS is headquartered in Cambridge, Massachusetts. That's where I'm based. Our single largest, fastest growing site is in Berlin, in Germany, and that's where the ML group is headquartered. And so overall, you see we're about 40 people, most of whom are in the US, but we also have 10 colleagues in Germany, plus students, 1 postdoc in France and in Marseille, and someone joining us from Estonia later this fall. And watch this space. Pfizer is very, very committed to growing our presence outside the US in research. And so I can't say exactly how our footprint will grow, but only that I know there's a strong commitment to grow our footprint outside the US and in Western Europe in particular. The other question I get is, what are you looking for when you are hiring entry-level scientists? And actually, most of our entry-level scientists are at the PhD level. Postdocs are not required unless it brings critical experience that's necessary to do the job that we're interested in filling. Drug discovery is the context in which we do everything. We do not expect scientists to come with drug discovery experience because that's what we'll be teaching you in this environment. We do expect training in one of the biomedical sciences. And so we expect you to have hands-on experience or formal training or both in one of the biomedical sciences. And then one of these other 2 skills, either applied math and algorithms or software engineering. Some people have all 3 when they arrive, but as collectively we have all 4, but and that's what makes this team kind of strong and unique. But in general, most people come with a scientific plus something else when they join our team. So to just give a high level of who we are and what we do, and I want to invite Joran up to give the first project example.
All right, thank you. So yeah, I'm one of the people that just started in Berlin. So in the new AI hub or the new location. So I just wanted to talk a little bit about a data loader that I've been making the last couple of weeks that might actually interest you. As you probably know, I don't know, is this my slide?
The next slide.
Oh, Eh? Wait. Okay. What am I gonna talk about here? Like, okay. No. Okay. I only recognize the first picture. Okay. Good. So as you know, there has been a lot of papers, people doing deep learning on epigenetics, ATAC-seq, ChIP-seq, all these kind of experiments, right? Very interesting, and there's a lot of papers, there's a lot of talks. Yeah, and of course, this is something we are also interested in at Pfizer. And in general, like what goes in is a sequence, and what comes out is either a binary, like a multitask binary classification of a peak being at a position on the genome, or you actually try to do regression and you try to actually predict the genomic tracks, right? And it has been shown. It has been shown that actually, if you do the last thing, that is actually working better. And I think I saw that as well in talks here this last week. But to do this, it's actually you end up like often, at least what I saw so far in a lot of the GitHub repositories, you end up with a lot of pre-processing. And I think one of the main reasons for that is that the package, the Python library that loads BigQuick files, where the genomic tracks are, is not really vectorized, and is not made to do like whole batches at the same time. So it's kind of slow. So this is exactly what we've been doing. We're trying to basically replace this pipeline by a pipeline where we do vectorize it. And one of the tricks, and I want to thank Andreas Pillman, who's one of the data engineers in our group, helped me with this. You can, for instance, do the decompression on a new NVIDIA decompression library on the GPU already, and this speeds things incredibly up. So this bar chart is really new. Just made it an hour ago. Basically, left, it's just like the scale. Don't look at the scale, but like it's using PyBigQuick, then if you just vectorize everything on the CPU, and on the right is if you do everything on the GPU. And this is, we will hopefully open source this soon, and then other people can use this as well. Thank you.
Yeah, hello, my name is Andreas Steffen. I'm also part of the Berlin team, actually, of Pfizer, and I'm working across cheminformatics, down to bioinformatics. In this case, actually, is one of the projects which bridge. So it's about mining the ligandable cystenome. And every now and then, actually, you get a data set where you're not just doing a one-off analysis and present a PowerPoint slide as a result, but you get a data set which is really of value across the projects which we are doing. And in this case, is about screening these covalently binding fragments. So these are fragments in the size of 200 to 300, all of which comprise a so-called warhead, which is binding, in this case, specifically to cysteines. And by using some proteomics measure and screening on cell lines and multiplexing and a proteomics approach, you can actually find where these fragments bind to and how strongly they bind. This is very interesting because it might provide you with very attractive starting points to tackle proteins in a new way. So there's also new modalities which do not require the inhibition or activation of enzymes or receptors, but which are essentially leading to the degradation of proteins. And then it's just actually important that you specifically bind to a protein no matter where. So in order to do some medicinal chemistry on top to optimize these fragments further, there needs to be an addressable proximal pocket. And what we have been building here is actually a pipeline in first place across the entire proteome. Where are addressable pockets, and what distance do these reactive cysteines have to these pockets? So for this, we were using the entire PDB, but also AlphaFold. Then you want to know, given a protein family, how specific is a cysteine compared to other members. There might be actually very interesting cysteines which are disease-specific, such as KRAS G12C, which you potentially could address through that. And then we also did some NLP actually to verify are there already existing cysteines which have been addressed using chemical matter. And we bring everything together and build COBE. COBE stands for covalent binders, but it's also a reference to one of the best basketball players, Kobe Bryant, which was covalently bound to his basketball. So our logo is actually showing that. But essentially, this is a visual analytics platform for our Pfizer chemists and structural biologists, or also biologists, which are doing target assessments. So for the deep structural exploration of these covalent cysteine binders. And our approach is really to focus on these candidate cysteines with druggable proximal pockets. And this is what it looks like, actually. So it's about integrative analysis of these data. So you can see it here. Let me see whether I have it quite enough. But essentially, there's a three-dimensional viewer showing AlphaFold and also superimposable X-ray structures from the PDB. And the second row actually shows all the cysteines which have been addressed. And as you click, you will see the chemical matter which is binding there. And also the viewer is focusing on the cysteine and showing the pocket or the proximal pocket close by. There's further data actually which we use for contextualization. For example, sometimes you might be interested in cysteines which are actually at a protein-protein interface, and we would show this. There's a second mode which is actually chemocentric, so you can search and find the fragments together with their data points, giving a certain scaffold. But we are also doing this multi-parameter ranking. So if you're interested in tackling any member of a protein family and you want to know which of the cysteines actually is most likely addressable and further optimizable from the fragment, then we're using this multi-parameter ranking approach, essentially using any feature which you provide, and you can actually make up your own scoring function to rank order the cysteines, and you can inspect all data from there. Such applications actually, as we are in a tech track, require quite a number of tools. And so there's, I mean, I would say the majority of work always goes into the data integration, the computation. But if you are not able to actually present everything to your customers, actually you're missing out, I think. It's not just an icing, but it's very essential for mining these data. And we're using, in this case, a lot of React.js and heavily this Molestar viewer. If there's anyone in the audience working with Molestar, I would be super happy to chat. It's quite a complicated but super nice package. And the visualizations actually we built essentially by rendering SVGs through this React technology and make heavy use in the background of D3.js. Yeah. So this is what it looks like. And please, any questions, you can also come to our booth.
Okay. So hello everyone, I'm Tuan, and I'm also working at Pfizer in Berlin in the machine learning hub. And today I'm going to talk about generative chemistry and how we are doing research here at Pfizer. And to first start with the problem formation, what are we interested in? Let's say we have a biological target, and then we want to design a pharmacologically relevant compound space with desired properties efficiently. And due to the large chemical space, enumerating the libraries or in general, like fragment-based approaches are computationally expensive. And recent advances in deep probabilistic machine learning enable us to train models that can learn distribution of molecular data. And the pioneering work, as shown on the upper right figure, is the work from Gomez-Bambarelli in early 2018, where they have trained RNN-based variational autoencoder on SMILE strings. So SMILE strings are string-based representations of a molecular graph, which should represent the topology of the graph. And they were training it in a variational autoencoder approach where an encoder network would process the string representation into a continuous latent space that has several nice properties where you can do optimization, interpolation, and also exploration. And once you have to do some optimization at latent space, you can use this the decoder network to also get back to the splice strings. And follow-up works also from one colleague of us, Robin Winter, he has been training a similar architecture on this kind of variational autoencoder approach, which is the CTDD model. And we have also shown that we could do efficiently same optimizations in the latent space. And my work now is, I'm going away a bit from the string representation, let's say, because if you have molecules, you could either embed them as molecular graph, or if they're embedded in 3D space, we have atomic coordinates. And the representation now that I'm dealing with is basically a point cloud, so a molecule embedded in 3D space. So we can say it's basically a triplet or a three-tuple. It has atomic coordinates P, vertices, which refers to the chemical element. And also we have the molecular topology, which is somehow the adjacency with all the edge features. And now we want to learn a probabilistic model that can, given a large data set, learn a distribution of such molecules embedded in this space. And a very recent probabilistic model, which has been shown very promising in the image processing community, that are the Noising diffusion probabilistic models, is that we could also use these learning algorithms for molecular data. And the general idea of diffusion models is that you're gradually perturbing your data using some tractable forward kernels into a tractable prior distribution, as you can see in the lower figure, where we have right now just in 1D kind of complex multimodal distribution, which would be our data distribution. And then we would have a tractable forward transition where we have samples from that complex distribution, which we usually just observe, which is then gradually going in time to a distribution which is tractable, which is on the right side, the prior distribution. This could be, for example, a Gaussian. And now how would this relate to molecules? Well, if we are looking now on the right, let's say we have a molecule that has some kind of like 3D conformer, or even actually in a set of conformers. And actually each molecule itself has a mostly very complex potential energy surface. And now assume we have a sample from that potential energy surface, which would be a data point or a sample on the left side, so from this complex distribution. And now we are using the forward kernel in order to gradually perturb the data sample. So that we are gradually destroying structure in the atomic coordinates. And the more and more we're going forward to this trajectory, we see that the point cloud, or at least the coordinates, they are squashing down to a prior distribution. So everything is centered around the origin, which would resemble the Gaussian distribution. So this is rather the how you would see it in a 3D molecular setting. Okay, so now that I talked about the machinery, so like the probabilistic learning algorithm, it is also important to think about, well, what kind of data we have, right? I told you we're working with sets, we're working with point clouds. Then it's also important what kind of neural network architecture we're using. If you're working in image processing, you're using convolutional neural networks. If you're working with sequences, RNN-based architectures have been prevalent and dominating, but now transformers are replacing them. But in general, if you're working with graphs or sets, let's say point clouds in 3D space, you're using graph neural networks. And essentially the idea is that you're utilizing the local graph structure through, for example, distances. You are creating radius graphs or K and N graphs due to proximity in 3D space. And then you're exchanging information between neighboring nodes. And also the richer the input is, for example, if you have 3D coordinates, the more expressive the output is also, because sometimes when you're reading machine learning papers, they always say the network learns it. Well, what does it actually learn? So therefore now here I'm showing you an example of how an iteration is processed. So now assume we have this molecule on the right, and we are looking at vertex 2, which is some kind of node, which is aggregating information from its neighboring nodes. So the embeddings are being learned through iterations. And let's say we are doing 2 iterations of aggregation. So we are breaking down the computational graph. So what does vertex 2 actually see? Well, it's easy. We just need to see how the vertex 2 is connected to its neighbors. So the one-hop neighborhood is consists of vertex V1, V3, and V4, while the 2-hop neighborhood consists of V5 and V9. So therefore, if we want to break down the computational graph, which is on the lower right, we see this is all the information which vertex 2 would see if you were to apply 2 iterations of message passing. How you define f theta is your own choice, but this is the general idea, like how you are in the end defining how information is flowing is important. And you can use graph neural networks or message passing neural networks for many tasks. In the machine learning and computational chemistry community, they have been used for supervised learning. And I'm just using here them now for as a means to encode that kind of data, so point clouds, in order to train a diffusion model. Okay, so now coming back to our initial objective, well, we want to train a diffusion model to generate or to learn a distribution of a large molecular data set. And now to recap, on the upper right, you see a more formal figure again, how diffusion works. As you say, you have a sample from your clean data distribution, which is in this case, this conformer. It looks kind of physically reasonable. There are no clashes. Let's say this is a sample of the potential energy service. And then you're gradually destroying structure in the point cloud by adding noise, right? And the forward kernel is tractable. And you know that at the end, on this trajectory, you have to run it for a discrete time step, let's say 500 time steps in each trajectory, the point cloud should be cluttered. So as if all the atomic coordinates were drawn IID from some Gaussian distribution in 3D space. And the idea is then to learn the reverse process. And on the lower right, you see an animation where it's this kind of learning methodology is applied in a, let's say, really de novo setting. So in this case, the molecular graph also changed through time. So we're learning next to atomic coordinates, also we are learning the chemical elements. So these are discrete data types, and there are then some technical details which we need to kind of follow in order to make the diffusion works. But this is a very interesting and promising scenario, what we can do. And this animation also shows some kind of unconditional sampling. So there is no context. And this is basically a simulation which you would do in gas phase, which is nice to study, but sometimes not so much of practical relevance. So it's more interesting to look into conditional sampling. So train a model that is conditioned on some properties to set some context. For example, you can think about if you were to do fragment-based design, you want to do linker design. So you have some fragments or scaffolds you want to preserve, and then you are basically learning a linker. And there are several works out there already, which you can find a citation down there, and they are basically using diffusion to learn molecules that are either able to design new ligands in the pocket, or that can preserve molecular structures. And I'm just showing you here now, for example, the stiff linker model, where basically if you had a molecule, you could apply your favorite decomposition algorithm in order to get fragments out of this, and then you can define a linker which the model should learn through diffusion. And what the authors have shown or seen in their paper is that if you are actually applying this model in a, let's say, unconditional setting, you are saying you have your target molecule, and then you have ABC, and A and C are your input fragments you want to preserve, then the model is still able to generate a plausible, reasonable final ligand. So the linker is still physically looks promising. However, they had problems to fit the ligand into the pocket. And this is why, because the model was not conditioned on the pocket. And this is also what you can see on the bar plot there, where the generated ligand, if there was no pocket information included in the diffusion process, the number of clashes increased, while if they were to condition the design of the ligand with the pocket, the clash is reduced. And then there are also other approaches where you can do de novo design in the protein pocket, fully unconditional. But the advantage is that, as I show you on the right side, so this is another paper, that you can actually train a diffusion model unconditionally. Maybe it doesn't make sense, but at least you are learning a very large probability distribution. But there are ways that you can adjust the sampling in a way that you can still preserve fragments or scaffolds. And this is also widely used in the image processing community, sometimes called in-painting, where you basically force the network doing sampling to preserve certain structures. And this is also what you can do with the molecules while training them unconditionally. Okay, then, yeah, thank you. And next to Joe.
Okay, so I think we have plenty of time here for me to go through the 3 slides that I have. So I'll take my time. So yeah, I'm Joe Salens. I've been at Pfizer for about 2.5 years. And, kind of what we've gone through today so far is moving along the drug discovery pipeline. And so there's a lot of early phase work going on, trying to find new targets. But then after that, there's also our final targets and the ones that we're trying to make and then put out into the clinic. And we also need support for those kinds of products. And so that's the kind of work that I work on. Specifically, the team I'm on is a team of computational biologists. We are contributing to helping to understand in depth the different nucleic acid therapeutics we have. So that includes basically the starting material for all of our biologics, so plasmids, which are, that's where everything that we end up making comes from. RNA, so the mRNA vaccines and other RNA therapeutics. And then also gene therapy, the viral vectors and everything that kind of goes along the way in producing them. And so let me just go to the next one. So yeah, our work really is focused on, it's applied toward the design of these molecules, so helping to figure out which one is best in a given bioprocessing condition. So we look across all these different ways in which these therapeutics are produced, made, stored, and then with what we really do to all these is we do a lot of sequencing. We have essentially every sequencer available that's somewhere in the company. So both next generation sequencers, a lot of Illumina machines, and then third generation sequencers, so Oxford Nanopore and Pacific Biosciences. Those are all tools that we use to help understand these different products. And a lot of these things you could maybe outsource, but we keep all of this in-house because as we get toward these later stage drugs, eventually we get these regulatory queries that come our way, and we have to have the expertise in both the products and the methods to be able to rapidly respond to these queries. And some health agencies, you only have a couple days to formulate a response and submit that. So we have to have all of this on hand, ready to go, and give our reply there. And so for all these different products, there's kind of a standard life cycle where we have the product, we have some feature that we're interested in understanding, so we sequence it in a way that will tell us the information we're looking for. So that's why we have these different sequencers, different methods to get to those questions. And then our group, it's our job to then come up with the customized bioinformatics solutions to answer that question. And then that goes back to the team. We understand a little bit more about products, so we keep moving along until we're happy with where we are. And so then I do have a poster. It's C112, so you can stop by, specifically on our work for supporting gene therapy. And I'll give you a little overview of what that is right now. So there are these recombinant adeno-associated viruses that are used for gene therapy. There are many different impurities that can come along for the ride as you're making them and getting them ready for the clinic. And as these are going towards patients and moving more towards clinical trials, there are many more. There's a much higher bar now for these standardized and systematic approaches to understand the capsid content of these viruses. And what we need to do why we need that is to, one, mitigate the vector toxicity. There are some products that can come along with these viruses that we have to account for and then reduce. And then our work, again, will also, in the process of getting all of these tools set up, that'll help us understand our products better, the processes that we're using to make them, and then optimize all of that together. And then, this is all because we have to really make sure that we're addressing the regulatory compliance for these drugs. And so I'm just going to give a quick overview of how gene therapy viruses are made. You can see these two, actually there's just one production system here. This is the HEC-293 production system. And so it's a very popular way to make these viruses. You have your transgene of interest encoded between these two inverted terminal repeats. You co-transfect with the minimum requirements you need to make that virus replicate. So rep and cap genes, and then the adenovirus components as well. You co-transfect those transiently into your HEC293 cells, and then you harvest your virus, purify, and now you have your gene therapy product. But there are, like I said, a lot of impurities that can come along the way, and these are things that we have to mitigate. And there are a lot of different approaches to understanding these impurities, but there's been one challenge that's remained, which is really understanding the variety of the impurities that are packaged inside these capsids. And we care about that, again, for the vector toxicity, but also making sure that we're having the most potent gene therapy drug that we could make. And so these are some of the impurities that we see. What we're trying to make is the little image on the far left, a full genome encapsulated inside that particle. But some of these product-related impurities, so those that can come through as you're trying to make this virus, include an empty capsid, so something that's not going to do anything, a truncated genome that is going to truncate your therapeutic gene, and then a chimeric genome. Sometimes you get this virus binding to host cell genomic DNA, or maybe even the plasmids that were coming along for the production. But then you can also see these other kinds of impurities, these product-related impurity, process-related impurities. So your plasmid might get packaged, your host cell genomic DNA might get packaged. And it's been a challenge for a long time to actually really figure out, like, what is the composition of these different libraries? And there's a really cool way that we've been addressing that, which is interrogating these genomes and impurities using single molecule real-time sequencing. And so we can, with PacBio, we can look at the full vector genome without fragmenting it. So now we can actually look at a single molecule level and see what the full content is of each molecule. And so this is kind of our setup for how we do this. We have our purified capsids. We extract the genomic DNA. AAV is actually will make a positive sense strand and a negative sense strand in equal proportion. So then what that allows us to do is we need to make a double-stranded molecule to be able to make our library. So we can just uncoat the virus and let those two opposite strands anneal to one another. Now we have a double strand. And then we can anneal the SmartBell adapters, which now allows us to have a circular molecule that we can then put onto the flow cell and generate our circular consensus sequences. And again, the real power of this is that now we're looking at a single molecule level, and we can see in the full context what that molecule looks like. And so now we have a way to actually address what are these different impurities in our library. And my poster is really just based on the lessons we've learned along the way that we want to share, both for PacBio sequencing and anyone interested in AAV gene therapy. So what we found is that there are, if you take just the recommended way of making a library and those that are recommended by PacBio, you might end up finding that this is not reflective of your the actual content of your library, because it turns out there are some biases in the sequencer, and I won't talk about those here, but we've done a very deep dive to understand this. And again, we have to, because this is all going toward regulatory responses. And we can actually see all the data we've been missing if we just follow the standard rules. And turns out we can actually see a much better representation of our library by kind of following our own intuitions based on what we've seen in the data. So I think our keynote speaker this morning was talking about the importance of really digging into your data, seeing what's in there. And that's something that we follow as well in our group. So we really do a deep dive to make sure we're understanding our products. And so, yeah, if you're interested in that topic or anything else related to this, you're welcome to come talk to me, poster C112. But I guess we can stop there and take any questions that you have.
Great job. We have 5 minutes.
You have
Yeah. A couple of plugs. First, great job, team, considering we didn't rehearse. That was awesome. Well done. So we are also participating in the job fair tomorrow, which is around lunchtime. I do think you have to pre-register. I don't think you just show up. So talk to the registration desk about what the process is. We'll also be staffing our exhibition booth, both in the morning coffee break and the afternoon coffee break tomorrow. And generally we'll have a number of us at lunchtime as well, if you have specific questions about the roles we have. I think that's all I wanted to say about the interactions with the team, but we do have 5 minutes, yeah, for any questions about any of the talks that you heard from previously.
So let's organize the questions.
Yeah.
There were one here. I don't know. Yeah, let's begin with it.
I've got a question to Tuan. I wonder, how do you decide how many hops of neighbors to include when you are doing the graph convolution steps in your autoencoder training? Or does the number of hops of neighbors actually affect your learning performance?
Yeah, so so in this slide where I showed you, if you embed them into the 3D space, the neighboring nodes are determined through a kind of cutoff. So you're computing, let's say, everything is connected within 5 or 7 angstrom, and then you will usually have local edges. And we have seen that at least for the diffusion models, that sometimes you would think you don't need that many iterations, let's say 5 layers, to kind of cover the entire molecule. But we have seen that even with even a higher cutoff, like many more layers have improved the model performance with respect to molecular stability. So like the samples we generated, they were preserving chemical valency, whereas it has been shown that previous models didn't have good molecular stability. And our model also right now learns the molecular graph topology. And the models before, they just learned the coordinates and the chemical elements. And yeah, it's still a bit of an open question in the machine learning community on why you sometimes need many layers, because in theory, if you're stacking many more layers, there's a phenomenon called over squashing. So basically you are exponentially letting all the interactions flow into a node. But at least for our use cases, if you have 3D coordinates, I think already having distances in the message passing gives you much more information, whereas if you were just operating on the molecular graph.
And could you add more context in your prediction steps? Because where you showed a case where you have a protein pocket as a context. I wonder, could you add more context environments to, in addition just to one?
Yeah, you can. I mean, right now, how it's mostly done is that you encode the protein pocket, you keep that fixed, right? So it's in the neural network, you would kind of have a masking token, but you can also include much more conditional information if there is a signal between the protein and the ligand. So it doesn't necessarily have to be protein structure like coordinates, but you could also use gene expression or something like this as into the model. So there's definitely more playroom.
Hi. Okay. Hi, about generative chemistry.
Okay.
I think when you use this methodology, we want to molecules to be synthesized.
Mm-hmm.
So how could you select molecules that can be good for synthesize? And so how could you select best molecules?
Yeah, it's a good question. So right now, as I showed you, it was purely unconditional, but in general, like if you were to do drug design, you would maybe use some kind of scaffold where you know they can be about in a library, or you know these chemical reactions are possible. And I think a very interesting approach is also maybe to include some kind of synthesizability model in the kind of sampling process, where you can then kind of give feedback into the sampling process and maybe steer into a direction of molecules that are synthesizable. So this is a very interesting direction one should also do. Yeah.
Yeah. So it's a bit of a question for all of the team.
how do you determine the skills that you will develop in-house and the skill that you'll kind of recruit out or even outsource?
It's a great question. I think we expect all of our staff to learn on the job and acquire new skills while they're applying the skills they already know, because I feel like the field changed so quickly. And so it's, everyone's best interest to continue learning and developing skills, attend conferences like this and other practical workshops. But when things need to be done at a certain scale, it's usually is like we'll be working with third-party vendors to do some of the upstream data processing, but we know how we would do it in-house. So we would define the steps and the quality metrics up front. So when they deliver the end product, we know if they deliver some of the quality like the way we would have done it. So that's how I would answer the second part of your question. It's related to scale. Okay, so that gets you to the Pfizer job board overall. You can see all our positions, not just the ones in our group. If you want to see the ones just in our group, you can go to the ISMB job board. And so if you filter on Pfizer, you'll see the 5 or 6 positions we're actively recruiting for in our group. So with that, thank you so much for coming.